Hello and welcome back to my YouTube channel. In this video, we'll discuss the apoptosis. So by the end of this lecture, you will be able to know the types of apoptosis, the morphological changes which are seen during the apoptosis, and the mechanism of apoptosis. So let's dive into the lecture. So, what is apoptosis? You must ask. Apoptosis is an energy-dependent process, or it is an active process, genetically programmed cell death involving single or small group of cells. Okay, so it requires ATP. So this was the definition of apoptosis. Now let's talk about the types of apoptosis. So apoptosis could either be physiological as well as pathological. So let's talk the physiological apoptosis first. So physiological apoptosis is involved in the removal of cells during organogenesis or embryogenesis. Okay, so removal of cells during organogenesis. Okay, so this was the first example of physiological apoptosis. Now let's talk about the next example. So we know that during inflammation, WBCs leave the blood vessels and enter the site of inflammation or infection. Okay, so after the completion of their function, they must be eliminated. Okay, otherwise there will be the exaggeration of immune response. Okay, so the removal of WBCs after the completion of their function occurs through the physiological apoptosis. Okay, so elimination of WBCs after completion of their function. Okay, so this was the second example. Now let's talk about the third. Okay, so physiological apoptosis occurs in the lymphocytes which recognize self-antigens. Okay, so during the maturation of lymphocytes, those lymphocytes which recognize body's self-antigens must be eliminated. Otherwise, there will be the chances of development of autoimmunity. Okay, so autoimmunity develops due to the defect in physiological apoptosis. So this example must be clear to you. So removal of lymphocytes which recognize self-antigen. Okay, so this was the third and the last example of physiological apoptosis. Now let's talk about the pathological apoptosis. Pathological apoptosis occurs during the chemotherapy and the radiotherapy. Okay, due to the damage of cellular nucleus. Okay, so due to the damage of cellular chromatin rather. So it occurs during chemotherapy or radiotherapy. Due to the damage of chromatin. This was the first example. Now let's talk about the next example. The pathological apoptosis occurs in the graft versus host disease. Okay, so it occurs in the graft versus host disease caused due to mismatched organ transplantation. Okay, so mismatched organ transplantation. So this was the next example. The pathological apoptosis also occurs in the virally infected cells. Okay, so virally infected cells are eliminated through the pathological apoptosis. The mechanism of it, we'll discuss it in the topic of inflammation or immune system. Right now, just remember it. So occurs in the virally infected cells. So now let's talk about the last examples. Due to the increased accumulation of mismatched protein, this mismatched protein elicits apoptosis. Okay, So increased accumulation of mismatched protein, or mismatched, not misfolded protein. Okay, so misfolded, rather we should say misfolded protein. Due to the accumulation of increased misfolded proteins evokes apoptosis. Okay, so with this we are done with the types of apoptosis, both physiological as well as pathological. So during apoptosis, some morphological changes are seen. So what all changes are seen during apoptosis? Let's discuss about them. Firstly, there will be the shrinkage of cell. Okay, so there will be the shrinkage of cell. This is the first event occurring during the apoptosis. Shrinkage of cell. Okay, so this is the first event. Due to the shrinkage of cell, the cell gets, or cytoplasm gets, more eosinophilic, or we can say it becomes more pink in color. So what develops? Hyper eosinophilia develops. Hyper eosinophilia develops due to the shrinkage of cell. Also, the chromatin material gets firstly condensed, and then there will be the fragmentation of that condensed chromatin. The former process is known as the pycnosis, and the later one known as the karyorrhexis. Okay, so firstly condensation of chromatin, and later fragmentation. So now we studied in the topic of necrosis that in the necrosis there will be the damage to the cell membrane, due to which certain inflammatory mediators were released, and these inflammatory mediators were causing the aggregation of inflammatory cells at the site of necrosis, due to which inflammation was occurring. But in this case there won't be damage to the cell membrane, so there won't be the inflammation either. Cell membrane remain intact. Cell membrane remains intact. So no inflammation. Okay. So this is the most important point you need to learn. Now let's talk about the last morphological change. Okay, so due to the fragmentation of chromatin, now cell divides into the certain bodies known as apoptotic bodies. Okay, so consider this is a cell. We know that this cell, firstly this is the normal cell. During apoptosis this cell firstly shrinks. The chromatin in this cell firstly gets condensed, then there will be the fragmentation of this chromatin. Okay, due to the fragmentation of this chromatin, now cell starts dividing into the certain bodies, certain membrane bounded bodies known as apoptotic bodies. Okay, so apoptotic bodies are formed. Okay, you must be clear about it. Now let's talk about the mechanism of apoptosis, and this is the most important part of this lecture. So apoptosis occurs through the two events, occurs in the two phases. Firstly, there is the initiation phase. Later, there will be the execution phase. Now let's talk about the initiation phase. First, initiation phase is further divided into the two pathways. First, there is the intrinsic pathway, and next is the extrinsic pathway. So let's talk about the intrinsic pathway first. Okay, so intrinsic pathway, also known as mitochondrial pathway. What happens during intrinsic pathway? Let's discuss about it in much detail. Stress, certain stress such as DNA damage, reduced growth factors, or hypoxia or metabolic stress activates protein known as BPH. Okay, so stress activates BPH protein. Okay, so due to the activation of this BPH, two events occur. Okay, firstly there will be the inhibition of BCL2. Okay, so there will be the inhibition of BCL2, which is normally involved in the maintenance of permeability of mitochondria by holding two pro-apoptotic proteins, which are box and back. Okay, so due to the inhibition of this BCL2, these box and box are set free. Okay, so normally these proteins are present in the inner membrane of mitochondria, but due to the inhibition of this BCL2, these block and box gets implanted into the outer membrane and makes the channel. Okay, so due to the formation of these channels, the permeability of mitochondria now increases. We know that in the intermembranous space of mitochondria, cytochrome C is present. Okay, so due to the increased permeability of mitochondria, cytochrome C leaks out. Okay, in the cytoplasm, cytochrome C combines with the APOF1 protein, forms the apoptosome. Okay, so this apoptosome now activates procaspase 9 into the procaspase 9. Okay, so this apoptosome now activates procaspase 9 into caspase 9, but there is problem. Okay, there is a problem. What happens? This apoptosome gets inhibited by protein known as IAP. Okay, which stands for inhibitor apoptotic protein. So we have to inhibit this IAP, otherwise there won't be apoptosis. So apoptosis won't occur. In order to inhibit this IAP, there is the release of certain protein known as SIMAC diablo. Okay, so SIMAC diablo is released from the mitochondria. This SIMAC diablo now inactivates this IAP. Okay, so due to the inactivation of this IAP, the apoptosome now set free, and this now can activate procaspase 9 into the caspase 9. Okay, this caspase 9 now activates the nucleases and proteases, thereby causing the apoptosis. So this was the intrinsic pathway. Now let's talk about the extrinsic pathway, which is also known as death receptor mediated pathway. What happens during the extrinsic pathway? We have got two receptors. Firstly, there is tumor necrotic factor receptor 1, and there is the FAST receptor, which is also known as CD95. When these death receptors binds with the death ligand, okay, so when these receptors bind with their ligands, there is the polymerization of adapter protein. Okay, in the cytoplasm, this adapter protein gets polymerized. Okay, so after the polymerization of these adapter proteins, procaspase 8 and 10 now gets activated into the caspase 8 and 10. Okay, so these caspase now can activate the nucleases and proteases, thereby causing the apoptosis. Okay, so this was the initiation phase of apoptosis. There will be the also execution phase as well, in which caspase 3, caspase 6 is activated, and these caspase 3 and 6 causes the activation of nucleases and proteases further. Okay, so now what happens to these apoptotic bodies? What happens to these apoptotic bodies? They must be eliminated. How are they eliminated? How are they recognized? They are recognized by the macrophages. How? We know that normally in the normal cells, phosphatidylserine is present at the inner leaflet cell membrane in the normal cell, this phosphatidylserine. Okay, but in the apoptotic bodies, this phosphatidylserine now flips to the outer leaflet. Okay, so this leaflet, so this phosphatidylserine now flips to the outer leaflet and then recognized by the epherocytes. These are the phagocytes in this case. Okay, so then recognized by the epherocytes. Okay, the process by which phagocytosis occurs known as efferocytosis in this case. Okay, so this must be clear to you. So the process known as efferocytosis. Okay, so the phosphatidylserine acting as eat me receptors, okay, or eat me signals. They are acting as eat me signals. There are also other compounds which are also acting as the eat me signals. They are called reticulin. So we could say that phosphatidylserine acting as the marker for apoptosis. Okay. So how these phosphatidylserine receptors are visualized? They are visualized through the annexin 5. Okay, so compound known as annexin 5. So they are visualized through the annexin 5. So with this, we are done with the apoptosis. We discussed the definition of apoptosis, which is the active process. We also talked about the types of apoptosis, physiological as well as the pathological. We discussed the all the morphological changes which are seen during the apoptosis. Okay. In the last, we discussed the mechanism and what happens to the apoptotic bodies, how the clearance of apoptotic bodies occur. Okay. So thank you for watching my video. Stay tuned.